Video covers the unique characteristics of the different classes of microbes and, more importantly, why we care about such things. So in this video we're going to cover the following topics: why we care about the defining characteristics of the different classes of microbes. Then we're going to go into what makes animal cells, bacterial cells, fungi, and viruses unique, and then we'll have a nice wrap-up summary. The learning objectives for this video are to make connections, so you're going to connect what you already know to the new material, and this also explains the interrelatedness of microbes that are adapted to living on or in humans, and later on we'll get into cultural nutritional practices. Well, first off, why do we care about the defining characteristics of microbes? Well, there's lots of reasons, but for this class, the two most important ones are these differences are the things that your immune system picks up. So if there's something that's unique to bacteria that never shows up in human cells, your immune system can pick up on it and attack it without ever having been exposed to it before. So important things to keep in consideration. Also, these unique characteristics are targets for antibiotics and other drugs, which will become important as we go along. Let's start out with what makes animal cells unique. Humans are animals, therefore these are the characteristics that make your cells unique. First off, your DNA is enclosed in a lipid membrane called a nucleus. We also have ribosomes. This is my little cartoon version of a ribosome, and ribosomes make proteins. And in eukaryotic cells, of which animal cells are a member of the eukaryotic cell group, it has an ADS ribosome. So don't worry about what this means, just it's a reflection of the size and shape of the ribosome. So here's how it works. A copy of the DNA is made. We call this mRNA. It leaves the nucleus, goes to the ribosome, and we make human cell proteins. And because we're animals, your eye cells make eye cell proteins, and your brain neurons make brain neuron proteins, etc., etc. Now, the other thing that makes animal cells unique is that there are no cell walls. So in other words, we have a cell membrane and then nothing rigid on the outside. Now, this is helpful to animal cells because it enables animal cells to ingest their food. So a white blood cell can swallow down a bacterium whole and digest it. For this class, the animals that we will be talking about will be humans, of course, and one type of worm, Trichuris suis or T. suis. Now on to bacterial cells. So here's my little cartoon of a bacterial cell, but let's make him bigger so that we can see what's inside of our little bacterium. Bacteria have DNA, but it is not enclosed in a nucleus. It is not enclosed in a lipid membrane. And they do have ribosomes. Notice I've made it a different shape because they are a different shape and they're a different size. We call them 70S ribosomes. Now, why am I having you learn the size of the ribosomes? Because this 70S ribosome is a target for some antibiotics. Something to keep in mind. And why am I telling you whether bacteria have a nucleus or not? Well, because that's one of the big divisions. Prokaryote, which doesn't have a nucleus, or eukaryote, which does have a nucleus. And also, let's talk about how bacteria make proteins. So once again, we make a copy of the DNA, and this is an mRNA, but you notice because there's not a separation between the DNA, the mRNA, and the 70S ribosome, the bacteria can start making bacterial proteins while this mRNA is being made. In other words, bacteria can grow and divide faster than animal cells. This becomes important when we talk about control of growth through either food preservation or antibiotics. Now, bacteria have a cell wall. In other words, they cannot ingest something whole smaller than them. And this cell wall is composed of a unique combination of sugars and proteins that we call peptidoglycan. Only bacteria make peptidoglycan. So when your body sees peptidoglycan, we're going to say sees in quotes because your immune system doesn't have eyes, then it can immediately attack, because we really don't want bacteria in our blood. Now, gram-positive bacteria have a thick peptidoglycan cell wall, and let's go ahead and get rid of that and show you what my cartoon version of a gram-negative bacterium would look like. It has a thin peptidoglycan cell wall, and then it has another lipid membrane on the outside that we call an outer membrane. This outer membrane contains a molecule called lipopolysaccharide, or LPS, which acts as kind of a super shock-inducing molecule, or an inflammation mediator in the human body. So that's something to keep in mind when we talk about gram-negative bacteria too. For this class, we're going to be talking about a lot of bacteria. A lot of bacterial species live on and in you, and so I have made a separate slide covering all of the bacteria that we're going to talk about. So here are the gram-positive bacteria. Remember, they've got a thick peptidoglycan cell wall. Here are the gram-negative bacteria. They have a thin peptidoglycan cell wall, that outer membrane that has the LPS. And then here are acid-fast bacteria, of which we're only going to talk about one, the causative agent of tuberculosis, or TB. And we're going to talk about that one more when we talk about antibiotics and also in the microbe of the week. Now let's talk about fungi. Fungi are your familiar mushrooms, mold that grows on your bread or in blue cheese, and yeast that make alcohol and make our bread rise and all sorts of good stuff. Now, fungi are eukaryotes, which means they have their DNA enclosed in a lipid membrane called a nucleus. Because they're eukaryotes, their ribosomes are ADS, which means that antibiotics that target 70S ribosomes are not going to bother them. In that way, they're very like us. But they do have a cell wall, but unlike bacteria, it is composed of chitin instead of peptidoglycan. So once again, this is something that your immune system can recognize, or it's a potential target for antifungal agents. We just haven't found one yet. The fungi that we're going to be talking about in this class are going to be Candida albicans and Saccharomyces cerevisiae, specifically the subspecies Boulardii. I think that's how you say it. Now let's talk about what makes viruses unique. So here's a eukaryotic cell. Here's a prokaryotic cell. You've noticed I've made them. I've made the eukaryotic cell about 10 times bigger than the bacterial cell, because that's the approximate size range that we see them in. And then here is a virus. Viruses are very, very tiny. We can't see them with a light microscope, with a few notable exceptions. And so that's one of the things that makes them unique. Also, they're not cells. They don't have a cell membrane. They don't have a cell wall. Basically, they're just DNA enclosed in a protein coat. Sometimes they do have a lipid outer membrane, we call it an envelope, that it picks up from its host cells. Viruses' main goal is to get inside of an actual cell. So I've shown this virus having gotten into a eukaryotic cell. We'll say that it's a cold virus that has gotten into you. And the first thing it needs to do is get rid of the protein coat one way or another. Then its genes, which are either DNA or RNA, start taking over the ribosomes of the cells. So instead of making a copy of the cell's DNA to make an mRNA, we make a copy of the viral DNA or RNA. It goes to the ribosome and we make viral proteins, which is used to make more viruses inside of the cell. Viruses are the ultimate parasite. They're not really alive. Now, why am I telling you this? Because viruses take over the cell machinery, takes over the cell ribosomes, and all sorts of other good stuff. Antimicrobials against viruses are very hard to make. Because if you kill the cells that have the viruses in them, that's not really good for the patient. They feel terrible. And so we try to attack things that are unique to the virus. So, for example, something involved in making the viral protein, something involved to keep it from ever getting into the cell. We try to target enzymes that are specific to the virus and that will leave the cell enzymes alone. That's some of the things that we need to keep in mind and why we care about what makes viruses unique. Now, for this class, the virus that we're going to talk about is herpes simplex virus. We're going to probably stick to herpes simplex 1, the kind that's generally associated with the mouth, although I'm sure you've all heard of herpes simplex 2, which is associated with genital mucosa. To summarize, here are the unique characteristics of the various types of cells that we're going to be talking about in this class. Animal cells do not have cell walls. They have 80 S ribosomes, and they have a nucleus. Bacteria have a peptidoglycan cell wall. They have 70 S ribosomes and no nucleus. That makes them prokaryotes. Fungi have a chitin cell wall. They have 80 S ribosomes. They have a nucleus, making them eukaryotes along with animals. Viruses are not cells. They have RNA or DNA in a protein coat, and it takes over cells. So let me know if you have any questions about the unique characteristics of various microbes. I'm happy to answer them.